So, welcome back. Mechanism of differential gene transcription. The protein-specific DNA binding sequence using chip sequence technology. Well, the chip is your chromatin immunoprecipitation sequencing. That means here what is exactly happened. The transcription factor which is bound or recognize a particular sequence in that promoter region of the DNA. It has affinity to bind a particular type of genomic sequence which need to be get transcribed. So by using this sequence, we can artificially create those proteins which is complementary to which can simply identify the particular set of gene which they are have affinity, and this and by this method we can simply have our desired gene easily. This is like your technique, uh, very nicely written somewhere. I can find it. Yeah, here it is. The technique that enables a researcher to use now the transcription factor as a bet that which we hold. We guess the pieces. A bet to isolate the DNA sequence that specifically recognize. The differential protein from high-low CpG contain a promoter. Uh, this high and low CpG content that play an important role. The promoter types are catalogued as having either relatively high or relatively low number of CpG sequence at the which DNA methylation occurs. So you can now going to read about the DNA methylation. In some of my previous videos I have explained about the histone methylation, but now here I'm going to discuss about the methylation at your cytosine residues of this DNA molecule. So in case of high CpG, this helps to regulate in this way. That means with if a CpG contain a promoter with high number of CpG isolate, multiple CG sequences there in the promoter also region, there is developmental control gene which regulates the synthesis of transcription factor and other developmental proteins used in construction of organism. So the default state here is important, that is your on, and they have to be actively repressed by histone methylation. That means they will be repressed by your active methylation. Getting my point? Not getting, I think. So let me once again clarify you in this picture. You better understand. Here you can see this is your high CpG content promoter. So the RNA polymers 2 can easily bind here, and its default is open type. But when there will be a poised state, the poised state is because of this methylation in this CpG cytosine residues, and that cause repression. And similarly in case of low CpG, what exactly happened? The low CpG content caused the transcription factor to prevent binding on the polymers 2, and that has caused the methylation of DNA, and there will be no transcription. So default is here no transcription. So the low CpG means genes whose products characterize major cell. So default state of this promoter is up, and they can be activated by your transcription factor. So high CpG, default is on, repressed by histone methylation, and here's low CpG can be default is off and can be activated with some transcription factor. Is mostly found in your developmental regulatory proteins, where this one is found in your some genes whose hormones are wait, yes, is product characterized in your mature cells, but here it is your developmental regulatory proteins are mostly concerned. Next video, if you want to know, next is your DNA methylation, switch on and off. So let me go through direct to the picture. Here one thing is the DNA is 5-methyl cytosine. That means cytosine get methylated. That's what I want to talk. So here's this picture. You can see in the fifth region 1, 2, 3, 4, 5. This is your CH3 group. That means methylated cytosine is there. So this methylation caused this. You can see this picture. Let me show you. This is the epsilon globin gene. There are different kind of globin genes are found during the developmental stages: alpha, beta, gamma, delta, epsilon. This kind of the epsilon and tau, these are found very early in the time where this produced are from yolk sac, rather than thereafter the uh, normal your what kind of gamma type of produced, and then after both this gamma type decreases and the alpha beta type remain high. So in this picture you can see the 6 week and 12 weeks there is a gamma globin which is active, but in case of 12 weeks it's inactive and your gamma globin becomes active. So this is done by your methylation of promoter region of the CpG acetylene of their cytosine. This is the thing. Now this is a very pictorial diagram. You can see here the easier transcription factor. I have told you this transcription factor can cause activation of the gene expression. So this is the activation is by done when the transcription factor bound, it causes the activation of gene, and when there is a methylated there is methylation, the transcription factor is unable to bind and thus cause inactivation of gene. So by removing the methylation we can activate the gene transcription, and by adding this methylation group we will inhibit the activation of the gene. That means the gene get inactivated. Here is the same picture showing a histone deacetylase. That means the steal group is removed. That means histone causing it's your repressed type. Here it is your histone methyl transferase, that means transferring that methyl group. Yes, it's transferring that methyl group. By adding this it prevents the transcription. That means it is also repressed. Both modification promotes the stability of the nucleosome and thereby tightening the packing of DNA, thereby repressing the gene expression in this region of DNA methylation. This is the two example. This is how DNA is methylated and repressed, or what you call deacetylase or repressed. Then here in this picture the methyl group you can see here is preventing the transcription to move around and cause gene expression. Okay, differential RNA processing we'll talk about in the next video. Thank you very much.